All right, I think my recording started, so I'll go ahead and get started. Again, for those of you who don't know, I'm Brandon Cook, PhD student at Clemson University, same place as Gabby, who just presented a couple of weeks ago, and actually under the same advisor, Dr. Duckett. Different from Gabby, though, we're going to talk a little bit about lipids this week. So to start off, we'll jump into it and just say, why are lipids important? Well, from a meat science perspective, I feel like they have a few important benefits, actually, and a lot of these take with a grain of salt with how they're typed, because it's actually kind of an increase in juiciness, increase in tenderness, increase in palatability. It's really perceptions of that. We know that as lipid content of meat increases, water decreases, so it's not actually an increase in real, true water capacity or juiciness, but it just gives that perception of more juice, more flavor. And as we talked about a couple of weeks ago with the various theories of how lipids improve palatability, we'll talk about that in the second lecture as well. But also fat is flavor too. You may have heard that before. That's probably why chicken tastes like everything, is because there's not much fat in there, whereas beef has a very distinctive fat flavor. And those steaks that Dr. Stelzlini likes and his wife doesn't, that comes from that fat on the product as well and how it's aged. What are we going to cover? Well, similar to how we started the semester, I figured we'd start this lecture series with how are fat cells made and how is fat itself made. We'll try and get a basic foundation of these topics before jumping into the second lecture, where I'll touch on how is the fat deposited, how does the meat animal handle it, and since they are meat animals, how does this fat impact the resultant product that we're going to use these animals for. So we'll jump into this first one here with how are fat cells made. As we can see at the top of the screen here, excuse me, we'll jump back here. Adipogenesis is the proliferation, differentiation, and conversion of cells into lipid-filling cells, or the creation of fat cells. Fat is going to come from the same part of that zygote of the neonate that muscle and bone is, and that's the mesoderm, as you remember. So mesenchymal stem cells are going to make that decision. Rather than becoming adipoblasts or osteoblasts or anything like that, they're going to travel down the adipoblast route, and they're going to become undifferentiated mesenchyme cells that will eventually result in adipocytes or fat cells. With some help, we can see here on the left-hand side, we go from the mesenchymal cell to this adipoblast. It's kind of rough on the edges, large nucleus compared to the cell size. This is going to be very proliferative. It's going to recreate itself and make a lot of adipoblasts, similar to how the myoblast is very proliferative. It's not a true myocyte. It doesn't have any of the proteins needed for shortening or anything like that, contraction. Similarly, the adipoblast is a fat cell, but it doesn't have any of the enzymes yet to make fat. It can't store fat yet. Its job is to be proliferative and make a lot of adipoblasts that will eventually start down the path of a pre-adipocyte. This is where it's going to start to round up. We can see in this next level here, it becomes a little bit smoother, still large nucleus, no fat droplet yet. But in this phase, it's just going to prepare to become a full mature adipocyte. You may have a few enzymes showing up, but nothing large. And then the final step, you get the final cue from the body that says, hey, I need a place to store energy, and it jumps into full adipocyte, where it's differentiated from the pre-adipocyte. It's fully rounded up now. There's major upregulation of the lipogenic enzymes. You may have had one or two, a few copies starting out in that pre-adipocyte, but now the production of those enzymes needed for lipogenesis has fully ramped up, and that lipid droplet we can see in this picture here, this one is very large, but again, that's its job, is to store fat. So, as I said, a whole bunch, there's all kinds of cues from the body as to where that adipoblast, pre-adipocyte, adipocyte, where that should be in its journey. Adipoblasts, again, are undifferentiated mesenchyme cells that are destined to be adipocytes but aren't yet. And then the next step is just to prepare. They're determined to be adipocytes in the pre-adipocyte form, followed by the adipocyte. And so where that happens, we can see at this adipogenesis at a glance, it's kind of a mess. There's a lot of things going on. I apologize, you can't read everything real well, but I don't know if many would if you could, because like I said, there's a lot of things going on here. We'll focus on a few big things as far as transcription factors. Those are going to be things that affect the cell to push it forwards through the steps of adipogenesis. And some of the main things we should think about, one is PPAR gamma. Many may have heard of that. That's just peroxisome proliferator activated receptor. We can see that here in this gold box, and that's going to play an effect as well as receiving an effect from CCAAT enhancer binding protein, or the CEBP right here. Additionally, retinoic acid is going to play a large part, WNT, and there's just loads and loads of molecules and transcription that act as transcription factors. Up here we can see that earlier on in an adipocyte commitment, that step between adipoblast and pre-adipocyte, IGF-1 acts on it, as well as about 10 other different things. So there's just all kinds of factors that play into whether or not a mesenchymal stem cell decides to be an adipocyte, and then from there, when it makes that step from a proliferative adipoblast to a waiting pre-adipocyte to the final mature adipocyte that has this large lipid droplet, as we can see down here. But like I said I wasn't going to talk too much about this, and probably already covered more than anyone will remember. So we'll move forwards, and I'll leave some stuff for that for further reading if you're interested. Adipocyte growth, then, once we get that adipocyte, how does fat tissue increase in volume, size, however you want to label it? True growth. We know that both hyperplasia and hypertrophy are methods of growth. Hyperplasia, to reiterate, is the increase in number of cells. Hypertrophy is the increase in the size of those cells. Muscle, we know that hyperplasia ends prior to birth. So whenever we're born, whenever the animals are born, the number of myocytes they have is set. You're never going to add another myocyte. They can only grow by hypertrophy or increase that size. Lipids are a little bit different. Lipids can both increase in hyperplasia through hyperplasia and hypertrophy. The number of adipocytes is not set at birth. And this makes sense because if you think about it, if you eat excess protein, the body breaks it down and gets rid of it as urea. If you eat a whole bunch of excess water-soluble vitamins, we're getting rid of that too. Fat-soluble, we have some issue with some storage that may be toxic, but the body doesn't like to get rid of energy. You know, if we're ever going to be stranded without food on an island, the body's going to start to break down the muscle to use the protein as energy, and that is the key thing. So the body makes sure that no matter how much energy we're taking in, we don't throw any of that away, or we try not to throw any away as best we can, which means that adipose size is going to change up until its maximum ability. We can see the cell diameter is ranged, we can see anywhere from 10 to 250 micrometers. And volume-wise, put that in perspective, the volume of a sphere is going to be the radius cubed, which, if you think about it, a twofold increase in diameter results in an eightfold increase in volume. So jumping from 10 to 20 micrometers makes a big change. But if you go from 10 to 250, you can think that's a huge increase in the amount of fat that is stored within that cell. But what happens if all of the cells we have reach that 250 micrometer size? Well, that's the cool thing about adipocytes, or in terms of human health, that is the very uncool thing about adipose cells, or adipocytes, because when we reach that maximum, the body just says, okay, we'll start splitting those mature adipocytes. It takes a little bit, splits that fat droplet in two, splits it both in each side, and it starts to regenerate more and more adipocytes until they're back down to small pre-adipocyte cell size, and can then start regenerating and refilling that lipid droplet up again. Now, how do we do that? Hypertrophic growth, we know, occurs through the accumulation of triglycerides, which is a glycerol backbone esterified to 3 fatty acids. They accumulate these triglycerides within the adipocyte lipid droplet, that big yellow circle we see in all those depictions of adipocytes. This happens in 2 different ways. The first is going to be incorporation of dietary fatty acids. It's going to be different between ruminants and non-ruminants. We'll cover that in the second lecture. But these dietary fatty acids, once we consume them, once they reach the small intestine, they're going to be absorbed, hit that portal blood vein, which is then going to go through the liver and be directed out to the the blood system, which then reaches the adipocytes and then enters into and eventually winds up in the lipid droplet. Now, the second portion of that, second method of increasing lipid droplet size, increasing the amount of triglycerides, is going to be de novo fatty acid synthesis. De novo just means from scratch. We're making fatty acids from the precursors or the excessive energy in the body. The secondary portion of this we're going to cover in a single slide is going to be the elongation desaturation as well, and we'll cover why in just a second. So to jump up here, de novo fatty acid synthesis is pretty simple. There's only 2 steps really, and acetyl-CoA is going to be our base molecule. This is going to be the messenger from the mitochondria that says, hey, we've got enough ATP made, we've got plenty of energy, let's start storing it. So acetyl-CoA is going to leave that mitochondria, hit the intracellular space, the cytosol, or depending on what type of animal it is, ruminants may just have acetyl-CoA acetone or acetate in the cytosol from dietary acetate. And that's going to be acted upon by acetyl-CoA carboxylase, which is going to be abbreviated ACC. And this ACC is going to take a CO2 molecule and add it to that acetyl-CoA, resulting in now a 3-carbon molecule known as malonyl-CoA. Now, malonyl-CoA then is going to be acted upon by a second enzyme known as fatty acid synthase, which will be abbreviated FASN. And this synthase is just going to take that 3-carbon molecule, chop off 1 carbon and the CoA, and add it to the backbone of the fatty acid. Now, it does this in 2-carbon increments. That's why the majority of the fatty acids you may ever see are all in increments of 2, whether that's C2, C4, C6, all the way up to C12, 14, 16, C18, 20, 22, etc. But we can see here C16, and there's a reason for that. And that's because de novo synthesis only can create fatty acids up to 16 carbons in length or less. Acetyl-CoA is the regulation step of this process. It's the highly regulated enzyme that is going to control whether this process is occurring or not. So even though you may only have a fatty acid of C10 that you're currently adding to, so you've got 2 more steps of this before it's complete, if energy drops in that cell, phosphorylation or dephosphorylation is going to occur on that acetyl-CoA carboxylase, and it's going to stop the process. However, we'll cover what happens then in the next slide, but the location of this synthesis is going to occur differently in different species. We can see that the pig is similar to ruminants, equine, that these well-known larger animals and or meat-producing animals, synthesis occurs primarily in the adipose tissue. However, you and I, along with poultry, true monogastrics, non-ruminants, no fermentation, very little fermentation in the digestive tract, the majority of fatty acid synthesis occurs in the liver. Now, interestingly, as I've said a couple times, it's interesting about fats that they can proliferate even after we're born. That's also a bad thing. Well, that bad thing comes back into play here, where obese humans, they've also started to see that obese humans can actually synthesize fatty acids in adipose tissue too, which may be leading to that excessive obesity as dietary energy stays high, but the expenditure of that, the metabolism decreases as we age. Now, back to that C10 fatty acid we left in the previous slide. What happens to that? Well, C10 can jump back into fatty acid synthase, but anything from C12 to C18, that can actually be acted upon by elongase 6 or ELOVL6, which adds, again, 2 carbons in length up to C18. So, for example, palmitate, which is the biggest fatty acid that de novo synthesis can create, can then be acted upon by elongase 6 to stearate. And in the same fashion, elongase 5 acts a little bit different in that it can work on polyunsaturated fatty acids. And it can carry those all the way up to 20 carbons in length. Now, we also know that if you've ever seen a lipid profile, there's not just saturated fatty acids. And in some animals, and most animals, the unsaturated fatty acids, they're not exactly like the dietary ones. So we're not just getting double bonds in our fats from the diet. And that comes into play with the desaturases. Now, there's several desaturases, 4, 5, 6, and 9. We can see listed at the bottom of the screen. But the very important part here is in yellow, where desaturases cannot insert past the N9 position. All right, so that means that omega-3 and omega-6 fatty acids that we hear so much about in popular media currently for health and that sort of thing, they can't be made by the mammal, can't be made in-house. They have to enter as a dietary fat and then acted upon these enzymes to alter them to what we need. So things like arachidonic acid, dodecahexaenoic acid, those come in through the diet, whether it be linoleic, alpha-linolenic, and are changed from there to what we need. Now, for anyone who's really interested, like I said, the adipogenesis precursors, the transcription factors that affect those precursors and how we make adipocytes, this Hausman et al. Paper from 2009 is a really good review on the biology and regulation of pre-adipocytes and adipocytes in meat animals. Covers a lot of stuff in detail. Also, if you go back to the slide with the adipogenesis at a glance, that book has a lot of detail as well and can tell you more than you ever want to know about adipogenesis. Now, as far as lipogenesis, there's been a couple papers. Most of the animal science is going to care about how dietary restrictions or diet or some treatment is going to affect lipogenesis. But Azen in 2004 has a really good paper, Role in Fatty Acids in the Adipocyte Growth and Development. That covers some details in lipogenesis. And Martin et al. From 73 covered some very early work on effects of fasting and how that affects enzyme profiles or the amount of those lipogenic genes and enzymes in the ovine liver and adipose tissue. So to conclude real quick, adipogenesis, the proliferation, differentiation, and conversion of cells into adipocytes. This includes the adipoblast, the pre-adipocyte, and the adipocytes, the mature adipocytes. Lipogenesis, on the other hand, is one portion of how the adipocyte fills the lipid droplet and stores energy. They can do so using de novo synthesis. However, this can only create fatty acids up to 16 carbons in length. However, the ACC and FASN that are used in synthesis, as well as elongase 5 and 6, and desaturases such as SCD, which is stearoyl-CoA desaturase, It's a delta 9 desaturase. These can change both de novo synthesis fatty acids as well as dietary fatty acids to be what the body needs throughout life. So with that, we'll jump back to this start screen. I'll make sure that Dr. Stelzlini has the website for this PowerPoint as well as the video you're watching, so that if you need to go back and write anything down, you can. If you have any questions pertaining specifically to something I talked about and want to get to the source, feel free to email me through the online learning community as well. So with that, we'll call this one an end and move on to the second lecture.